Can you hear me on this one? Oh, yes, you can. Oh! And I can hear it. Okay, great, great.
We're doing the online people. Well, first of all, thank you to so many people who took time out of their summer to come and join us today to celebrate this lady here. I'm Jamie Cornelius. If you don't know me, I am Jalyn's advisor. And the plan is that first I will introduce Jalyn and try not to cry. And second, Jalyn will tell you about her research, during which I expect she won't cry. We'll probably have some acknowledgements where she'll try not to cry. And then the crying will be done, and we'll have questions for a period of time for some questions. And then the committee will take Jalyn into the interrogation room and talk to her about her research. And hopefully then we will soon after celebrate a new doctorate. Okay, I wrote down what I wanted to say. It's less than 5 minutes. So when I first met Jalyn, I had advertised an NSF-funded master's position when I was still at Eastern Michigan University. And very quickly, when you advertise nationally, received at least 50 applications from really good, excited undergraduate students. And Jalyn, though, was one of the first to contact me multiple times as a signal of what was to come. And she stood out right away. She had a 4.0, she had intensive field experiences, glowing letters of recommendation, and she rose to the top. She was also a local girl, being from Ohio. And so I thought she was likely to be quite happy in Ypsilanti, Michigan. And in fact, her sister had been at EMU and really enjoyed it. So this all felt like the stars aligned. And a few months later, I was going to meet her for the first time on her way to Ireland. Sorry.
No, I met you in January.
Earlier than, okay.
And then I went to Ireland.
Okay, a little later, second. So I was meeting her for the first time, funny enough, in my house. I just finished building. And this swirling fireball of energy came swooping in, and very soon was regaling me with her theories about the universe. Maybe have had that happen. And her place within it. And I remember thinking a few things after that first meeting. One was, holy crap, what just happened? Two was that this girl is on an intense personal journey. And three, wow, what am I? And what a powerhouse of a human. I felt both trepidation and excitement over working with her. And not for the last time wondered if she should really be pursuing a PhD in philosophy. It would be easy to say that Jalyn is fearless, but that would suggest that she is also careless and thoughtless. And I've never met a person before who is more thoughtful than Jalyn. I have the utmost respect for her ability to self-reflect and self-criticize and grow. She has pushed through some really hard, redefining moments in her life. She did. And some repeated setbacks in her science. From being my first PhD student at OSU and suffering a steep learning curve at my side, to the disruptions of COVID, to the inevitable frustrations of studying a nomadic songbird. Just one example is a whole month that we spent together in the Olympic Peninsula, trying to catch the hundreds of crossbills that were gleefully flying around in the canopy, and we could not get them down to the ground. That was field season fail number one, and it's not her fault. Jalyn has come out the other side of these challenges a better person, a better teacher, a better scientist, and one hell of a first student. That's the point where I might cry. Jalyn has been a huge part of my success here at OSU. She has cultivated the proactive, supportive, problem-solving, positive team culture that exists today in our lab, and for that, I will be forever grateful. I think she's also built that more broadly in our department, and she's often taken personal risks to be a voice for graduate students. She was not fearless in those actions. I saw and heard many concerns and tears and fears over the years, but what will always stand out about her is that she always did what she thought was right. She did not take the easy way. She is so very brave. All right. Jalyn is leaving a legacy that extends far beyond her science, which she'll get to shortly. And that's something that I'm certain she will do wherever she goes in life. She's already dreaming about how she's going to combine her intimate knowledge of anatomy and physiology and endocrinology with her passion for running ridiculous distances to create resources for women and non-binary runners. To see a student graduating with that kind of energy is really rewarding. And again, I'm so proud of you. I know it wasn't easy, but you produced a really solid dissertation. You got two kind of scary strong feet on the ground. And I can't wait to see what you do with all of that courage. So without further ado, I'm very happy to introduce Jalyn and let her tell you about the science that she has done while here at Post.
Nope.
There we got it.
Okay, it's good enough. Okay, great. So my dissertation is titled "The Impacts of Weather and Season on Reproductive Physiology in an Opportunistic Breeder, the Red Crossbill Loxia curvida." This is my dissertation defense. So I wanted to start it with a quote from the Fellowship of the Ring. It's dangerous business, Frodo, going out your door. You step onto the road, and if you don't keep your feet, there's no knowing where you might be swept off to. And I would like to send you through the journey of the last 6 years of the places I've been swept off to, and this isn't even half of the journey because this is just a little snippet, and I've had multiple field seasons where nothing happens. So this is just a little snippet of where I've been swept off to. So my talk breakdown today will start with an introduction on just simple avian annual scheduling and the breakdown of these different life history strategies throughout an annual schedule. And then I'll break down my 3 chapters that are moving through a crossbill year, talking first about winter breeding, moving into the spring migratory transition, and then finally going into the fall to talk about molt and this breeding hiatus that crossbills generally take. And then at the very end, I'll tie it all up with some conclusions. Great. So we'll start with an introduction. So most birds partition the year with these different life history strategies. And if you're not familiar with what that means, I'll break down these different life history strategies to give you a little bit more of an idea. So this all starts with an overwintering phase where birds start the year somewhere in the wintertime. And then they typically take a spring migration where they end up on a breeding grounds where they can breed. And then they molt before again coming back to migrate in the fall back to their overwintering grounds. That's a general overview of an avian annual schedule, moving through these 5 main life history stages. This takes place where we see a lot of seasons, and those are going to be in the temperate zones. And I'm going to focus my talk today in the North Temperate Zone, where we are right now. And so photoperiod is just changes in the day and light cycle throughout the year. And in the North Temperate Zone, we see the largest fluctuations in daylight, where we see an increase in the amount of daylight during the day to the summer solstice, and then we see a decrease in the amount of daylight all the way until the winter solstice. And at that same time, we see increases in temperature that coincide with these increases in day length, and then as day length decreases in temperature. Alongside with this change in photoperiod, change in temperature, we see changes in what's available for food for birds. One of those things are the green up of vegetation throughout increasing day length, increasing temperatures. We also see insect emergence and abundance there in the spring as well. So more food in the springtime. Not only does that change the environment, but changes in photoperiods stimulate bird brains. So light comes into a little bird head, and just for funsies, not really relevant here, birds have 3 different places that they have photoreceptors. So they have photoreceptors in their eyeballs, in the pineal gland, and then in the hypothalamus. So light comes in and it stimulates the brain. And that then, don't worry, turns on these biological clocks inside the brain. The first one we're going to look at are called circadian rhythms. Those are just day and night 24-hour cycles. This also stimulates longer rhythms called circannual rhythms, and that's where we're going to be looking at longer term physiological and behavioral changes through things like reproduction and migration. So let's look at that bird brain photoperiod stuff. So light comes in, stimulates the brain, turns on these clocks. That's sending information into the brain that then sends information to the endocrine system. So this is a neuroendocrine response. Brain information, then sending it to the glands of the body. So for the duration of my talk, I'll be focusing on the adrenal gland through the hypothalamic pituitary adrenal axis. And then I'll be focusing on the gonads, the testes and the ovaries, and the hypothalamic pituitary gonadal axis. Right. So the hypothalamic pituitary adrenal axis, or the HPA, I'll have for the duration of the talk labeled in red. This is going to be initiated by some sort of environmental perturbation. So something that stresses a bird and makes that bird need energy. We might also see this when there's some sort of energetic need like molting, where there's a lot of energy needed, more energy needed to the body, more HPA response. So I'll have this little lightning bolt here. There's some sort of environmental perturbation that tells the brain to start this hormonal pathway. So there's this cascade of hormones from the hypothalamus to the pituitary gland to the adrenal glands. And what we care about here is the response, the production of corticosterone, which is the bird HPA stress hormone. In humans, we often see this hormone called cortisol. And what we really care about here is that this hormone, when it's secreted, makes energy available in the body, and it does that through the breakdown of proteins, through the production of glucose, and through the breakdown of fat. And all of this happens within 3 minutes. So when we take a bird out of a net, we have to catch it really fast and get a blood sample so we can get a baseline level of how much hormone is in the blood without us stressing the bird out by grabbing it from the net. So when we get that baseline sample, we get it within 3 minutes. And then if we get it anything after 3 minutes, we call that stress induced. So we'll see that later throughout the talk as well. So when we're looking at CORT, we see how this changes throughout these circadian or 24-hour cycles and these circannual cycles, these longer term physiological changes. So through that day and night cycle, we see that CORT goes down during the daytime and it goes up during the nighttime. And that makes sense because at nighttime you're not eating, so you need some sort of fuel in your body. So you're going to start producing CORT to get glucose in the blood. We also see the reactivity of the HPA axis change throughout the annual cycle. So this is that stress-induced response of the HPA axis. So if we're looking at something like molt, the literature shows that that reactivity is actually suppressed. There's less reactivity to the HPA axis. And sometimes in reproduction, we actually see that HPA response can be higher. Which then moves us to the hypothalamic pituitary gonadal axis, or the HPG axis. And you'll see this in blue. So this is going to the testes and the ovaries. So we already learned that photoperiod stimulates the brain, and that's what initiates this pathway in birds. And the first hormone that's secreted is from the hypothalamus, and it's called GnRH, or gonadotropin releasing hormone. And I highlight that here because we're going to see it again throughout the duration of my dissertation, because I will actually inject birds with this hormone to see if the gonads can respond with the production of testosterone. So hypothalamus produces GnRH, and then downstream effects, that's going to produce the sex steroids, testosterone, estrogen, and progesterone. Okay. We also see that the HPG responds differently throughout these life history stages. So for example, here, we're looking at males on the x-axis, and we're looking at the amount of testosterone on the y-axis. And we see there's actually a lot more testosterone in the system when males are practicing laying and building nests. So laying eggs and building nests. But we see it's much less when they're not breeding or when they're doing other portions of reproduction. And it's really cool to study both the HPA and HPG axis because the literature shows that they have an inverse relationship. Higher levels of CORT start to suppress the HPG axis and result in less testosterone and sometimes even nest abandonment if birds are in the process of breeding at that time. Sweet. All right, so we talk about photoperiod initiating all of these life history stages, but there are other kinds of environmental factors that are then stimulating the bird to then move into and fine-tune these life history strategies. And so some of those factors might be things like how much food is in the environment, what kind of temperatures that bird is experiencing, what types of precipitation are at that time of year, and then how much water is available in the environment. And so we can even just think of how changing environments could then influence the amount of food that's available, the amount of water that's available, what temperatures exist. And so it's really important to understand that climate change is dramatically changing the Earth at a rapid rate. And it's now becoming a point that we can no longer deny that these things are fluctuating. There are changes in temperature. Those changes in temperature result in changes of water availability. and changes in precipitation that then result in changes in what food is available in that environment. We also see that in the US alone, by the end of the century, we're going to have dramatic increases in temperature pretty much across the board. And those changes in temperature result in dramatic changes in precipitation with areas of extreme drought and areas where we're going to experience way more rainfall than snow. And just in the Pacific Northwest alone, we're expected to see potentially up to 10 degrees Fahrenheit increase in annual temperatures. And by the year 2080, we're expected to have little to no snow left in the Pacific Northwest. And that's because we'll see this transition of snow to rain during the winter season. So birds have adapted this really long evolutionary scale of time, have adapted these optimal windows for things like breeding, things like molting, things like migrating back to their overwintering grounds. But as we shift the environment and we start to change what food is available or change what water is available, we might start to see these mismatches in when birds then participate in these life history strategies. Birds that are constrained by season, that have adapted these really long evolutionary life history stages, might be stuck breeding at the same time of year. But if they're breeding at a time of year where the food is no longer the same, it might shift then what happens in the next life history stage. So we might see that breeding takes longer than normal, but that then could expedite molt and then result in poorer quality feathers. Or that might mean that molt is pushed into a longer portion of the fall, where then those birds are migrating at a later time of year, and that's going to then cause a mismatch in what food's available when they're migrating. So it's really important to study how these animals shift their life history stages throughout different climate conditions. Which then brings me to my study species, the red crossbill, because these are considered an opportunistic species. And that just means that they theoretically can breed anytime food is available for them. So you can see here this figure on the right, crossbills have been seen to breed pretty much any time during the calendar year, except maybe those later fall months of October and November. So crossbills forage almost exclusively on conifer cone seeds. And so when they find a cone crop, they sit on it for the duration of the year and eat those cones. And as long as the food's good, theoretically, they can breed. So it's really cool to study a species like this because they aren't technically constrained by these really specific seasonal patterns. So we can see how environmental conditions might shift their phenology and then what that might mean for birds that are seasonally constrained. On the left-hand side, I show that these birds are sexually dimorphic. So red crossbills get their name because the males are presumably red. Males can also be what I call confetti birds, where they're orange or yellow or orangey yellow or orange, yellow, and red. Sometimes they look like the females, but classically the males are red and these females are this olivey kind of yellow-gray color. And then their babies are streaky and cute and brown, and we can't sex them without doing genetic testing unless they start to show some of their feathers coming in as like red or orange. Okay, that's it. So let's talk about crossbill annual scheduling. So we'll move into the winter season. This is going to be my first experiment, and then we'll move into some more, in a way, observational studies throughout the annual cycle. So my first chapter is looking at how temperature and precipitation affect reproductive and energetic physiology in red crossbills during the winter season. So my advisor, Jamie Cornelius, and then her advisor, my grandpa of science, Dr. Tom Hahn, they have over 30 years of observing red crossbills in the wild, and something that they observed during the winter season is that birds in the wet coastal regions of Washington tend to breed less frequently than the birds in the really dry and really cold regions of the Grand Tetons in the wintertime. So that made us then think, why are they doing that? How do temperature and precipitation affect winter breeding in this and energy allocation in this opportunistic species? So that led us to this hypothesis that cold, rainy winter conditions are energetically costly and they inhibit or delay reproductive development in red crossbills. So to test this, we took a bunch of birds in the lab and we separated them into two treatment groups. We had a warm treatment group at 21 degrees Celsius and a cold treatment group at 6 degrees Celsius. And in each of those groups, or in the warm group, we had 8 birds in dry conditions, 8 birds getting rained on in the lab. We had 16 birds in dry conditions and 16 birds in the cold getting rained on in the lab. And then this all began in October. And we started putting birds in mixed sex housing pairs, and you can see this little purple box around our birds. This purple box is a soundproofing chamber because of the environmental chambers have this loud sound. So to mitigate that sound, we put them in this box. We moved them into these places for 2 weeks to give them time to acclimate, and then we started raining on birds. Let's see if I can get this to play for you. All right, so we rained on birds for 30 minutes a day, 6 days a week at random times. We did this for the whole duration of the experiment, which was about 3 months. It was very wet and very icky in that room, and poop gets wet. It's gross. Okay. Anyways, so throughout this experiment, we bled birds at 3 different timestamps: late fall, early winter, and late winter. And then in early winter and late winter, we started to conduct those GnRH challenges. So remember, GnRH is that hormone that sets this cascade of effects to release testosterone. So we were seeing if the birds were actually capable of producing testosterone at that time. We also took video scores of these birds to see if they like to hang out near their neighbor or not during rain trials. We did that in early and late winter, so a behavioral study on their affiliative behavior. And then at the end of our experiment, we sacrificed the birds and looked at all their gonads. And then I went into the lab and I looked at the hormones using hormone assays to see how much testosterone was there and how much corp was there. Other measurements we took were seed intake rates. So we measured their food every single day. Shout out Adam, wherever you went. Yeah. We measured their food. And then we also did other body metrics like looking at muscle, mass, and fat. And then we also scored those behavioral videos, seeing how often they were spending or how much time they spent near their neighbor versus on the other side of the cage during these rain trials. So our predictions here were that cold and rain conditions would increase seed intake rates. They would need more energy. This would increase baseline levels of CORT, needing more energy. This would increase the response of the HPA axis to capture and handling. They would have lower levels of circulating testosterone. They would have less of a response to that injection of GnRH, and their gonads would be small. So at the end of our experiment, here we go. All right, so looking at males first, on the x-axis, I have season, so it's late fall, early winter, and late winter, and on the y-axis, I have the amount of testosterone in nanograms per milliliter. And then a reminder, induced testosterone is the response when I inject these birds with GnRH to see if the testes are responsive to that hormone. And what we found was that testosterone did increase seasonally. We see that increase in baseline testosterone from early winter into late winter. And then we do see that the birds had more testosterone after the injection of GnRH right here. We see that is also greater than baseline. So they had more of a response with the injection of GnRH, and then it also increased from early winter into late winter. And this follows what we see in the wild with red crossbills starting to breed in February into March. So this kind of follows what we expect to see in wild populations. For females, we didn't have the ability to look at estradiol because the assays require like 30 to 50 milliliters of blood plasma. And that's a lot of blood for a little bird. So we can't do those hormone assays on these females, but we looked at ovary wet mass. So on the x-axis, we actually see temperature. Season did not have an effect here. Well, I guess we can't because it's the end. Never mind. And then on the y-axis, we see the ovary wet mass in grams. What we found, though, is that the females that were in the warm conditions had heavier ovaries than the females in the cold conditions. And that's indicating then that there might be some sort of energetic cost to being in these cold environments in the development of the gonads. But you can also see these are pretty small. 0.025 is grams is very small. So these ovaries were still not creating follicles. They weren't vascular. They weren't in any sort of reproductive condition. Looking at all of our little video trials, seeing how much time birds spent near their neighbors, we have percentage of time near the neighbor on the y-axis, and then each of the bars is the individual birds from the experiment. We see that those birds housed in dry conditions spend a lot more time near their neighbor than the birds housed in the wet conditions. So this is another maybe more evidence to suggest that these rainy conditions are then changing the behavior of the birds for this more affiliative behavior towards reproduction. We looked at food. So on the x-axis, we see every week of the experiment. On the y-axis, we have food in grams. And we saw that birds housed in warm and dry conditions ate less than all other treatment groups. That's what this red bar is at the bottom. And then statistically, we see that birds housed in cold conditions ate more than birds housed in warm conditions, and birds housed in rain conditions ate more than birds housed in dry conditions. So that's another piece of evidence suggesting that rain and cold temperatures are then some level of energetic costs making birds eat more food. And then the last piece for this chapter is our friendly HPA axis. So on the left-hand side, you see baseline CORT. On the right-hand side, we're going to see induced CORT. The season had no effect on CORT. And the only parameter that mattered for our baseline levels of CORT was actually precipitation. So precipitation's on the x-axis on the left here, and nanograms per milliliter of CORT on the y-axis. And we see that birds housed in rain conditions had higher levels of circulating CORT as opposed to birds in dry conditions. So again, more evidence that these rainy conditions are some level of energetically costly. What's interesting though is our induced CORT on the right-hand side here. We have temperature on the x-axis and induced CORT on the y-axis. And what we see is birds housed in warm conditions actually had a higher HPA response. And what we think is happening here is that either these birds housed in cold conditions are fulfilling their energetic needs by eating more, as we saw before, or that they have change their metabolic machinery in order to cope with these colder conditions where the warm birds haven't had to do something like that quite yet. All right, so let's finish the winter season. Some conclusions here are that we're seeing that rain and cold conditions are imposing some level of energetic costs on red crossbills. We saw that through CORT, saw that through the amount of food they're eating, saw that through their behavior. There might be some trade-offs that are evident that are hindering females from developing their gonads as quickly as males. And that there's something about those warm conditions that were easier for the females to invest in the development of the gonads. But it's also, there's so much bias in the literature because it's really hard to study female hormones. That's another place that we can invest more energy in female development for reproduction. But we're also seeing more and more evidence that there is a level of seasonality for this considered opportunistic species. And we see that with those males increasing testosterone throughout the season, regardless of those climate conditions. All right, let's move to spring. It's already 30 minutes in. Okay, moving to spring. All right, so let's talk about the spring migratory transition in crossbills. So this was more of an observational study and watching the birds change in their physiology over time. So there are lots of different migratory strategies in birds, and the classic migratory strategy that many of us think of are birds moving north and south for their breeding and overwintering grounds. So on the left-hand side, we have the example of the blackpoll warbler, and this is considered an obligate migrant. That's obligate means this bird has to go north to breed, south to overwinter. So these birds move north, they're going to breed, and then eventually they'll come back down south where it's nice and warm during the overwintering season. And that's your classic obligate migrant. Another migratory strategy is called a facultative migratory strategy, and that's where birds are either exploiting some sort of resource like red crossbills, where they exploit conifer cone seeds. They find a good conifer cone crop, they eat it, they stay there for a while, and then when that food source is depleted, they leave. It's called an evasive strategy. When there's no more food available, they'll up and go. And so we see little conifer cones. These birds no longer have food. They don't have food anymore. So now they're going to go migrate around until they find a good conifer cone crop to settle on. And those are called facultative migrants. Because crossbills forage almost exclusively on conifer cone seeds, they follow typical patterns of conifer tree masting. So conifer trees produce seeds usually between May and July, and then those crossbills will also migrate between May and July, trying to find a nice cone crop to settle on for the next year. And so this led us to then ask the question of, do these facultative migrants transition to migratory conditions similarly to obligate migrants? And the literature shows that Carduet lionfinches, or the subfamily of crossbills, they all kind of change their fat throughout the annual schedule differently, but these two nomadic species on the bottom, pine siskin and red crossbill, they change their fat slightly more similarly. And if we zoom in on crossbills in the springtime, we see that they increase fat as they approach that migratory state when they're trying to find conifer cones. And once they settle on those cones and start breeding, they drop their fat again. So we're going to look at that. Oh, yes, but also, nobody has ever studied sex hormones in these birds through this migratory transition. So we're going to see if we can find the same patterns, but then are they preparing to reproduce once they get to their conifer cone crop? So to do this, we had colleagues at Washington State University that have outdoor aviaries on the rooftop of their biology building. So we used their colony. And we watched these birds across the spring season from April to early June into late June. And so at each of these timestamps, we took blood samples. Here we just took baseline cort and baseline testosterone. We also looked at fat, at muscle, and their gonads. And then at the very end of all of this, I again did hormone assays to see how much testosterone was in the blood, how much cort was in the blood. And here we predicted that as these birds are reaching migratory transition or migratory condition, they're going to increase fat, they're going to increase muscle, flight muscle, and increase baseline cort. A lot of the literature shows that birds rely on some level of cort for migratory timing. We also are predicting that birds are getting ready to breed once they get to that conifer cone crop. So we're going to see increases in baseline testosterone and increases in gonad size as we go through these months. All right. So as we're looking at fat and muscle, fat and mass, on the x-axis, we have the season, the sampling session. So it's April, early June, and late June. On the y-axis on the left, we're looking at what's called residual mass, which was really our size corrected mass for the bird. So it just means all of our birds were then corrected to be a similar size. On the right-hand side, we're looking at fat. And again, we expect to see both of these increase over time. So for mass, we see that they did increase from April to early June, and then they kind of plateau into late June. But what's interesting here is that we see that the females were actually increasing mass earlier and more than the males. And so this could be because females produce eggs, which are going to be heavier than producing sperm. It could also be that these females are preparing to migrate faster than males. And then looking at fat, we see a similar pattern of an increase from April to early June, and then another plateau into late June. But what's interesting here is we have birds that were caught as juveniles and birds that were caught as adults. So all these birds by experimentation had been in captivity for over a year. So these are all adult birds at this time. But birds that were caught as juveniles are starting the season with way less fat than birds as adults, or that were caught as adults. And we think this could be some novelty to these birds never having the opportunity to migrate or breed in the wild. So they might be developing at a different rate than adult birds. And we'll see some different patterns with these juveniles later too. Funky little birds. We expected to see mass and fat increase, and we also expect to see a correlation between mass and fat. And we do see in general that as fat increases, we see increases in mass. And then looking at muscle, there was no change across the season, but again, we see this kind of funky situation happening with birds that were caught as juveniles versus birds that were caught as adults. And here we see that birds that were caught as juveniles and male had lower fat scores than birds that were caught as juveniles as females, and all birds that were caught as adults. So again, this is showing that there's some level of developmental difference between juvenile caught and adult caught, and that there could be a difference between sets. There you go. Here, I want to shout out my undergraduate student, Dimitri, who ran all of my core assays for this experiment, and then also all of my core assays for my next chapter. So thanks, Dimitri, for being a super good technician. Here we're looking at cort again, and this is just baseline cort. So how much cort is circulating in the blood without any sort of stressor. So on the x-axis, we have the month. So April, early June, late June. Y-axis, we have cort in nanograms per mil. And we see that cort did increase across season, again, from April to early June. And then we see this sort of plateau. But again, we see this weird interaction of sex versus season, where females actually ended with higher levels of circulating cort than males. And this could be, again, that females are preparing to migrate at a different rate than males. It could also be, there's a lot of literature that females in captivity have a harder time breeding than males. Another bias that's in the literature with captive experiments on birds. And so it could be that females are just having a harder time transitioning than males and they need more energy. Maybe that's dumping it into eggs. Maybe that is that it's just harder being a female in captivity. Okay. We also looked at baseline testosterone across the season. And again, we expect to see increases because as these birds are going to approach their new conifer cone crop, they're going to start breeding. And again, on the x-axis sampling session, April, early June, late June, y-axis, we see testosterone in nanograms per mil. And we see there's basically no testosterone in these birds. That dotted red line here is the limit of detection of our hormone assay. And we see that only 4 birds were above that line. So even though we have data points down below that line, we can't actually say the concentration of testosterone is accurate there. It's so low that it's not detectable. So these birds did not have a lot of circulating testosterone. They were not in a reproductive condition. But what's interesting is when we look at the gonads. So let's take a look at those. On the left, we have the testis length. On the right, we have ovaries. So left is males, right is females. And again, we have sampling session on the x-axis. So for males, what we found, looking at their testes, is that a lot of these males fall above this line here, and this line shows how, the spermatogenic length of testis. So if bird tests or crossbill testes are above 4 millimeters, they're considered spermatogenic and capable of producing sperm. So all but 4 of our data points were above that line. So all these birds had big enough testes to produce sperm. However, they weren't producing testosterone, so these birds were not actively reproducing, but they were ready to as soon as it was time. For females, we see the ovary score did also not change across season, but none of these birds had follicles that were developed enough to reproduce. So if they were a score of 6, that means that they would have yolking follicles that are ready to go. They weren't quite at that state and they did not change across time. But in both of these scenarios, now we have a flipped situation with our juvenile birds. So the birds that were caught as hatch years, those dashed lines, we see at the onset of this study in both groups had larger gonads than the birds caught as adults. So even though their development in this more migratory transition seemed delayed compared to birds caught as adults, these birds seem like they're investing in reproduction earlier, even though by the end of the study, they were not statistically different. All right. So looking at that spring migratory transition, you see that birds increased fat and mass as well as baseline port following what we see in obligate migrants. So these facultative migrants are transitioning into the spring migratory transition similarly to obligate migrants. We again see that there's some sort of difference in males versus females, and we see this as the males had testes that were ready to reproduce at any time, where the females were still not quite at a reproductive state. We also see that those females were laying on mass at a faster rate than the males. And then we also see that those juvenile caught females were laying on muscle more heavily than the juvenile caught males. And then we also see these weird differences in juvenile birds that it could be that there's a novelty there that they've never had the experience of breeding in the wild or of migrating in the wild. So they may have slightly different developmental timing than adults, and that could be a direction people could go for some further research. Right. So then we'll move across the summer. We'll skip the summer because they almost always breed then, and we'll move into the fall where it's cool. Okay, so let's look at the bird crossbill molt and breeding hiatus during the fall season. And I want to focus this time at Mary's Peak. So Mary's Peak is just a little southwest of Corvallis, if you saw it on your way into town, if you're not from here. And Mary's Peak is the traditional homelands of the Kalapuya people. And they call this mountain, I want to say it correctly, Chatimanwi. And this is a spiritual place for the Kalapuya people. And today the living descendants of these people are part of the Confederated Tribes of the Grand Ronde and the Confederated Tribes of the Siletz Indians. So I want to really thank Mary's Peak for existing. It's been an awesome place. And I caught my 3rd data chapter there, and then we caught 146 birds that season. So I'm really grateful for Mary's Peak existing. And it's a beacon of hope when you're driving into Corvallis because you're almost home. Okay. So in the fall of 2023, the Oregon Coast Range was an optimal place to be a red crossbill because lots and lots of different conifer trees were producing cones. So moving left to right, the trees that were producing seeds were Douglas fir, Sitka spruce, and western hemlock. So if you were in the Oregon Coast Range during that fall, you heard millions of crossbills. And we know that those birds were breeding when we got there in September because there was a ton of juvenile birds flying around. Yeah. So we wanted to look at this fall season, and we see, based on this figure here, that crossbills don't seem to breed very often in October and November, and we wanted to know why and if they're actually capable of breeding during this time. One thing we know that they do during the fall is molt. So you can see here, the most intense phase of molt in the red crossbill is during these fall months. And molting is very energetically costly. And if you're not familiar with what molt means, there are lots of different phases of molt. We have integument molt, so you can see really flaky skin. They're replacing their skin. We have body feather molt, where they're replacing all their body feathers. And then we also have primary molt, which are these outer wing feathers. So replacing all of these different parts of the body increases metabolism and takes a lot of energy. So presumably, as they're molting, this could be a reason that they're not investing in reproduction at this time. So we want to know, is this opportunistic breeder actually capable of breeding during these fall molt seasons? And so this figure here is from eBird. eBird is a citizen science database, and where there are really dark purple boxes, that's where there's a high abundance of birds. And we can see right here, Mary's Peak, dark purple, lots and lots of crossbills. So I took Jessie. Hey. Took Jessie out to the field and we put up mist nets. If you've never heard of a mist net before, it's this really fine mesh net. You can barely see it because you don't want the birds to see it. You put up this fine mist net and we had decoy birds, live decoy birds in a cage because crossbills are super social. So having this mist net there, crossbills come down to see who these people are, birds, and they get caught in our net. And then we have to really quickly get them out of the net. And then remember when I said the HPA axis happens really fast, happens within 3 minutes. So I got to get a blood sample from that bird within 3 minutes. Once we get that blood sample, we have baseline cort, baseline testosterone, and then I can inject my bird with GnRH. Again, GnRH sets that pathway into motion for the testes to produce testosterone. And then we put our birds in a bag for 30 minutes, so then we can get that induced level of cort and induced level of testosterone. And then after we get those, we then take measurements of our birds. We take other body measurements like muscle, fat, mass. We put a band on them, and then we let them go. And then I take all that blood back to the lab, do hormone assays, and see how much testosterone was in the blood, how much cort was in the blood. Other measurements that we took that we haven't seen yet are external reproductive measurements. So for males, that's called the cloacal protuberance. So birds, like reptiles, they only have one hole for reproduction and for excreting things, and that's called the cloaca. So when males are reproductive and producing testosterone, they will elongate the cloaca so it can easily get sperm into the female. For females, we can look at brood patch, and a brood patch is basically a little blister on the belly that fills with blood plasma and can keep eggs warm on the nest. So we measure those. So for cloacal protuberance, we can measure the length of it. For brood patches, we can see, is it feathered? Is it not feathered? Does it have vascularization? Is it filled like this? Or is it wrinkly and it's done? So that's how we score those things. All right. So some predictions for the fall season, we were predicting to see an increase in mass as they move out of the breeding season and start investing in molt, increases in molt, and then increases in baseline cort. As we move away, remember HPA and HPG axes are working against each other. So as we move away from the HPG, increase in the HPA. We are expecting to see decreases in baseline testosterone, decreases in induced testosterone, and then we would see that the CP, the cloacal protuberance, and the brood patch would go down over the season. So looking at males, again, we have testosterone here and cloacal protuberance. So on the left, we see testosterone across months. On the right, we see cloacal protuberance. So testosterone basically had none from the onset. So there was very low levels of circulating testosterone from September, and then it stayed the same. Even with GnRH injection, there was no response from the testes. So we didn't see an increase in testosterone even after we injected them with GnRH. So these males are not ready to reproduce and they're not producing testosterone. And we saw cloacal protuberance was still long in September, but already by October was back down to non-reproductive levels. And this is a secondary sex characteristic. So this shows that HPG was already downregulated on the left. And it just takes a little bit longer to get those secondary sex characteristics to follow. But immediately we're back down to non-reproductive levels by October and into November. So for females, the only measurement we have here is brood patch. We couldn't take large quantities of blood from these females because they're actively working with their babies. So we don't want to do that. We see that brood patches were existent in September and immediately not existent come October and into November. So these females were again no longer reproducing. They were because they had brood patches, but they're not now. Okay. For molt, we expected to see molt during the fall season because that seems to be a regular pattern for red crossbills. And we see that their body molt was high in September and was finished by November. And then primary molt, which if we look at this little pine siskin over here, Cardueline finches have 9 primary feathers. So a 0 means no primary molt. A 10 means they're done molting. But if I were to say it's a 7, that means the 7th primary feather was molting. And we saw that birds molted all their primaries and were completed by the end of November. So both metrics of molt occurred during the fall season. And then coming back to our friend the HPA, I want to thank Dimitri one last time for doing all of my court assays, and Dimitri is officially the court king because he was so good at them. And we expected to see increases of court throughout the fall, and we did in fact see increases in baseline court from September to October. And then it plateaued into November, and we see increases again in that stress-induced HPA response from September to October, and it plateaued. So a similar pattern from baseline to induced across the fall season. For mass, we expected to see an increase in mass post-breeding, and we actually saw a decline in mass across the season. And anecdotally, Jesse and I were seeing a lot of seeds falling from the conifer trees at Mary's Peak during really dry spells. And so it's possible that these birds didn't have enough food to eat by the end of the fall season or were getting ready to facultatively migrate away from Mary's Peak. And again, anecdotally, if you looked at eBird from fall into spring, there were much less birds at Mary's Peak. Is that good grammar? I don't know. There were less birds at Mary's Peak from fall into spring. So we did see that birds did leave. And so this HPA response could be a response to molt, and it could also be a response to getting ready to migrate away from Mary's Peak. And this decline in mass could be that when these birds are molting, they're putting more blood into the body to invest in those feathers. And then as the molt goes away, there's less blood in the body. It could also be that these birds didn't have enough food and were getting ready to leave. All right, so some conclusions here. There's some level of a limiting factor in either the anterior pituitary or in the gonads to GnRH. The birds did not respond at all to GnRH, so there is no response. We see that crossbills do regularly molt during the fall season, and it was completed by the end of the fall season. And we see that the HPA might be important in molt, but it also might be important in preparing birds to leave in this more evasive migrational strategy. Well, good on time. That brings me to my conclusions. So I want to have a little reminder of photoperiod being kind of the initiator for a lot of these life history stages. And then all of these supplementary cues of how much food is available, how much water is available, what temperature a bird experiences, and what type of precipitation is there are all working together to fine-tune life history strategies in birds. And in my first chapter, we looked at these 2 supplementary cues, temperature and precipitation, across the winter season. And what we found was that birds in cold temperatures increased the amount of food they ate, and birds in cold temperatures had smaller or less heavy ovaries. Birds experiencing rain ate more food and had higher levels of circulating corticosterone, but we also saw that birds in rain conditions spent less time near their neighbors. So there is some level of these supplementary cues impacting the amount of energy allocation and investment in reproduction in red crossbills. In chapter 2, we didn't manipulate any of these variables, but we did see as birds are reaching this migratory transition, they're increasing fat, increasing port, and increasing mass. But we can know that as environments are changing, we're going to see changes in things like what food is available and what temperatures a bird experiences and how much water is available in the environment. We can expect to see changes in investment in these different life history strategies moving towards migration that might actually shift these optimal times of when birds breed and when birds then migrate. And those shifting strategies will then have later downstream effects that could either expedite other phases of the life history stage, or it could then push things outside of optimal windows. So if we push breeding to be either longer or outside of its optimal window, it could then affect things like molt that we see with our birds in the fall. So which brings me to my final chapter again, where we saw increases in court and increases in molt. And if those things are pushed outside of their optimal window, that could then impact when crossbills are migrating or when crossbills are molting, and then impact how they can thermoregulate in the winter, or impact if they can leave if food isn't available during the fall. So what we saw, where there are going to be changes in food availability, temperature, and precipitation during the fall season, and that does have deleterious effects on species that might force them to facultatively migrate or leave where they are and find better places with more resources in order to survive the winter or invest in breeding. So crossbills are kind of a unique bird where they're not constrained so much by photoperiod like our friend the Blackpoll warbler or classic obligate migrants that are moving north and south. But they're not immune to changes in the environment. So even with just, this is from the Audubon Society, even just increases in 1.5 degrees Celsius, we see that crossbills lose 35% of their range. And then even with an increase of 3 degrees Celsius, now crossbills lose 59% of their range. So even a bird that we consider resilient and flexible will be impacted by increases in temperature, and we will see increases in temperature. We're already seeing those increases in temperature, and gymnosperms are what we see with conifer trees. And these are already changing when they're producing seeds, where they can survive. And that's going to then shift where crossbills can be and where crossbills are foraging. We're seeing the hottest temperatures on record. There's a ton of climate projections that are showing snow melt, and when snow is falling as rain, we're going to have changes in how much water is in the soil, how much water is available in snowpack, how much heat we can keep on the planet through snowpack. So all of these changes are not just impacting birds, but they impact people too. And people in the Pacific Northwest like us typically like to recreate outside. So if you care about recreation and don't care about birds, you should still care about climate change, because again, by the end of this century, there should be little to no snow left in Oregon. And if you like to ski, this is going to be Mount Hood, and you're going to have the teeniest little bits of places to ski. So even if you don't care about crossbills or other songbirds that are going to be affected by these things, you should care for yourself. Okay, so with that, I want to thank my committee, Jamie, Bob, Doug, Brenda. Thank the office staff, because man, we wouldn't have gone anywhere without these people. They helped us schedule our classes, figure out where to go. I really am thankful for Tara, because she helped me start the C2C fundraising run. She was part of me getting in touch with Dee to get that going. All these people are essential. You can read it in my dissertation. Okay, I want to thank everybody that made me a teacher. All of the people that helped me as an instructor, the people that teach the classes that I teach, but also 2 TAs, Jesse Laney and Dan Schneck. Both of these people were mentor TAs for vertebrate biology and for anatomy and physiology, and they taught me how to teach and how to teach those classes. I want to thank the Cornelius Lab. I was the first one. It was just me and Jamie at the very beginning, but look how much we've grown. Yeah, we've gone to conferences together. We, wear Halloween costumes and do all that good stuff. Okay. I want to thank the C2C. It's an awesome run. We've done it for 3 years now, so you can see how it's changed and grown over time. Katie, you've run over half of it with me, so thank you. And you can see that it's changed to where we're starting to incorporate mountain biking, but what's pretty incredible is that we've raised over $8,000 in the last 3 years for undergraduate research and experiential learning for our students. Which we all benefit from, and I've taken a couple in the field that have had some of those funds. Thank the greater IB community. Is it gonna go? No! Thank all of the graduate students that make grad school fun. You can see that we have lots and lots of activities that we partake in, and many of them seem to be outside. All of these people kept me sane for the last 6 years, and I'm super grateful for that. Thank you. You can see it was just a lab of 2 in September of 2019. This was at Ann Devonsong's wedding. If anybody cares. But Jamie has shown me a lot. She has literally watched me grow up. As just a little 23-year-old, and I've changed so much since then. All right. Thank Dot and Kenny. We're there. Dot and Kenny have been my home away from home since I moved to Portland. They're my house in Corvallis. This was when Dot had to leave me, and it was COVID, so we couldn't say goodbye to each other. Hi, Kenny. I didn't know Kenny that well at that time. And thank yeah, and thank their cats, the most cuddly, needy girls of all time. Wait for it. Gonna go. Oh no! Wait, Jessie. Jessie was forced to be with me for a long time, and I think at first we weren't sure we liked each other, and then we had to like each other, and then we became like sisters where we fight. But at the end of the day, Jessie has become one of my closest friends because she had to. But we've been all over the place together. We've spent months, we've spent 2 field seasons over 2 months long in the field together. We went to India together. We've gone to so many conferences together. Had a long journey. And I'm not crying, which is awesome. Okay, and then I want to thank my greater community that's not just graduate school, but greater Corvallis and Portland community that also kept me sane, supported me through some crazy endurance events, and we just had a lot of fun outside together. Thank my family who is here today. Traveled all the way from Ohio to see me defend my PhD. Yay. Okay. And then last but not least, I want to thank my partner of 5 years for supporting me financially, mentally, and emotionally to get to this point. Yeah. And then I will end with a quote from Octavia Butler. All that you touch, you change. All that you change changes you. The only lasting truth is change. God is change. And questions? All right. Russell.
Fantastic.
Nice.
So talked a lot about like early on how photo period really influential for birds in general and in a lot of different aspects. And you kind of talked about how it seems in some of those aspects like migration not be influencing Red Cross bills. But did you at all manipulate photo period in like your experiments, or did you think about how photo period like in your lab might be different from like the outdoor lab aviary at WSU, like how those things might be affecting the production of on the any of that?
So when we were doing our captive experiment, those birds were housed at the Corvallis photo period, but also if you saw, we had double the amount of birds in the cold treatment groups. So those birds were at a British Columbia photo period, and then we had another group that was at the Corvallis photo period, and there was no difference in most of our metrics. Jessie might have some, no. There was no difference. There was no difference in most of these metrics. There's slight differences when it comes to the ovaries, but our sample size is so small that those differences were like birds of 3. 3 is not a large enough sample size to really be biologically meaningful. The Washington State birds, they're just barely a higher latitude. So there was not a lot of a difference there, but all these birds were moving in a natural photo period, a natural progression. So we weren't seeing a difference in when that onset was happening. Yeah. Okay. Yeah.
On I'm just a sibling of a scientist here, but
Of Ayana?
yes.
Okay.
You said multiple times that they specialize in conifer. Yeah, so, and then later on you said that they came to Mary's Peak because of various other seed pods.
Mhm.
However, none of those were conifer. Was it the sheer abundance of seeds that brought them there, or?
Those are conifer trees, so those are all different
Oh, I'm so
types
sorry.
of no, it's fine. Why would you know that?
Okay.
Yeah, there are lots and lots of different types of conifer trees, and all of those, like what we call evergreen trees with cones, are conifer trees.
Oh,
So they're
okay.
foraging on pretty much, not all of them, but a lot of different conifer trees that produce seeds. That's what crossbills like to eat.
Okay, thanks.
Yep, that's why they have a cute little bill. Yes, Giovanni.
All right, Lake Mathis asked via Zoom, did you measure wing chord or chord? Is there much variation in wing length that might relate to increased migratory efficiency, resulting in decreased stress response for more efficient flyers?
Can I read Dr. Mathis's question? That was a long question. I'm gonna look in the chat.
Yeah.
Is it in the chat? No, it's in the Q&A.
Q&A.
Okay. Did you measure wing chord? Oh, we did measure wing chord. Is there much variation in wing length that might relate to increased migratory efficiency resulting in decreased stress response? We did not look at that, but that would be something interesting. We do have the data for that, so that could be something that happens later. We could look at the wing length and relate that to their levels of baseline chord and then even induced chord. But we did not do that for my dissertations. If you,
Do you want me to add something to that? Because, so we,
you want to speak in the microphone?
oh, right.
It's capturing her voice, so.
Oh,
It's
cool.
okay, but I don't.
You can ask what you say.
There's one published example.
Oh, there's a published example of that, Dr. Mathis. Anybody else? It's 2:00. Russell?
Did you ever figure out why the red coffee goes from yellow in captivity, and did they turn yellow at Washington State?
I don't think we've ever determined what exactly is making them red versus yellow. It's something about diet, we think.
You're not feeding them purely wild conifers.
Correct. We feed them sunflower seeds and then rowdy bush, which is just a nutritional, like, wheat-based thing. But there is someone at Auburn that's trying to figure it out in house finches, which are a closely related species. Still not quite known what is making yellow to red in pregnant finches. Yeah.
Well, great presentation, Jalyn. I was very happy with it. You did great. But how did you inject the hormone and where did you inject it?
In the pectoral muscle. So we have little syringes and then we like move the feathers away and we put it in and squeeze it. Yep.
Cool.
Dave?
This is probably way outside your area. I'm
Probably.
just curious, you know which photosensor actually triggers the HPA and HPC actions?
I mean, I don't know for certain, but it might, but because they have photoreceptors in the hypothalamus, I have to speculate that then that's going to initiate the hypothalamus to produce GnRH and secrete GnRH. Crossbills for funsies, they do produce GnRH year round, they just don't release it year round. So it could be that they just need something to stimulate that release. Yeah. Giovanni,
Another Zoom question.
okay. Should I read it?
Yeah.
Okay, for the temp range study, were the neighbor birds always of the opposite sex? Do you think there could be some kind of pair versus peer dynamic? As in, if the birds are energetically drained, maybe they are only investing in social energy and potential mates as opposed to peers. We tried to put them, we put them all in what we morphologically said, and from capture were male versus female, but we had one group that was 2 males. Yeah, and those actually had some of the largest testes of the entire group. So we do think that there might be some then competitive interaction with those 2 birds being housed next to each other. But it is possible that these birds, you know, this proximity to neighbor is just a way that we can look at behavior, but it's not a surefire way of saying like, yes, this bird likes this bird because they're in a house together. So yes, it could be that they're drained. It could be that they're just really social. And there is some anecdotal evidence that maybe the male versus male was making bigger testes.
Yay.
Thank you all for coming. You're all a good part of Jalyn's journey, and thank you for being here for the end of it. Dot has one more